Great. Okay. We'll start over. Good morning, everybody. Thank you for joining us for Cardiology Grand Rounds. It is my great pleasure to introduce Professor Jen Davis, who is the director of the Center for Cardiac Biology and the associate director for the Center for Stem Cell and Regenerative Medicine here at the University of Washington. She is a world-renowned expert on cardiac fibrosis, myofibroblasts. Particularly as as a non-expert in that space, I really enjoyed the title of a recent talk you gave called "What is a Myofibroblast?" which I feel like might be an appropriate place for some of us, though I know we also have a lot of other experts in the audience today. So thanks for joining us, and thank you for joining us this morning, Jen. Take it away.
Okay, well, first I wanted to just start by thanking Greg for the invitation, and it's really great to see some of you guys in person. So just to start out, hopefully I can get this slide to go. Okay, there we go. So for those of you who don't know me, my lab's been interested in understanding the basic tenets of fibroblast biology. And I think along the way, we've really started to uncover some of the cellular and molecular underpinnings of the heart's fibrotic response. And so why should we care about the heart's fibrotic response? Well, we know that it's pretty central to heart failure. In fact, it's the replacement of healthy muscle tissue with fibrotic scar that often drives the progression to failure in the heart. Oh no, now it's advancing. Let's go back. Okay, so one of the things I wanted to point out is that fibrosis is really easy to visualize. You can see that with the simple histologic stain that's shown here. So on top, you can see healthy mouse heart tissue where all the healthy muscle cells are stained with red. But if you look at an infarcted mouse heart, which is shown down below, you can see that most of that healthy tissue is replaced with a blue stain. And that stain is actually fibrotic connective tissue. Oh, goodness. And so while I've shown you histology sections from an infarcted heart, fibrosis is actually a part of most etiologies of heart disease, and those are shown for you here. So there's fibrosis associated with cardiomyopathies, hypertension, coronary artery disease, aging, and so on. And so I really think it behooves us to understand the basic process of fibrosis. Okay, so one of the holy grails of heart failure research has been to discover a way to keep fibrotic scarring low in order to maintain as much healthy active heart muscle as possible. And this is actually a quote from Eduardo Marban that was put in Time magazine about 10 years ago now. And some of the approaches that have been used to sort of address this holy grail of heart failure research are illustrated here in this schematic. And so there has been a lot of work in pharmacologic approaches to reduce fibrosis. And most of those have turned out to be ineffective long-term. And we think they've been ineffective because we didn't understand the mechanistic basis for the process itself. Another approach that's been taken that I think is a pretty interesting approach is to reprogram non-muscle cells into heart muscle. And it turns out that happens to be pretty low efficient or have low efficiency, but also the delivery method for those reprogramming factors are non-specific. And it's still unclear when you wipe out a non-cardiac muscle cell population what the ultimate downstream effects are on the heart's physiology. So I think there's still a lot of open questions on that approach, and whether it's going to become a bona fide therapeutic is unknown. And so two other approaches that I think UW is really good at and have paved the way in is they're more regenerative medicine approaches that are revolving around cell therapeutics. So either delivering de novo cardiac muscle to the myocardium or implanting cardiac tissue patches. But one of the problems with this therapy is that it still has to overcome the barrier of a fibrotic scar. And so oftentimes these delivered cells or these implanted tissue patches get encapsulated by a fibrotic scar, and then they can't integrate with the host and they can't electrically couple. And so this still sort of sets up a premise that we need to find some way to address that fibrotic scarring. And so just to give you some background, the work from my lab and others have now really identified, I think, the cell type that's responsible for fibrosis, and that's a cell called the myofibroblast. And so for those of you who don't really study myofibroblasts, this is what they look like here in the center in two-dimensional culture. And here they're stained with alpha smooth muscle actin, which is shown for you in green. And you can see that alpha smooth muscle actin in these cells incorporate into these like really beautiful stress fibers. And once that happens, these cells can actually increase their ability to contract. And that's important for, say, for wound healing functions in multiple tissues. Another characteristic of the myofibroblasts is they become positive for a matricellular protein called periostin. And in fact, the expression of periostin happens really early in the process of developing these cells. The other primary hallmark of a myofibroblast is that it secretes a large amount of what we call a protective fibrotic scar. And again, that's shown for you here in this Masson's trichrome stain in blue. And again, this is an infarcted mouse heart. And so if too many myofibroblasts form or their activity is too great, the scar can actually take over a large part of the volume of the heart. If too few myofibroblasts form or their activity is impaired in some way, the scar can fail to form, but this actually puts your heart at risk of rupture. And so there's a delicate balance of how much you want to really play with these cells in order to address the fibrotic response. So I think one of the more important features of a myofibroblast is that they don't actually exist in healthy tissue, but they're derived from a local source of fibroblasts, so they're resident to the heart. These fibroblasts come from the TCF21 and PDGF receptor alpha lineage, and they sense injury or stress, and then they undergo a programmed differentiation into this new cell type. And so one of the places where my lab has come into trying to understand this process is to really understand the molecular drivers of this differentiation between a fibroblast and a myofibroblast. We've also been interested in, understanding how plastic this transition is and whether or not it's reversible, because you could consider like this might be a really nice area for therapeutics. And so to try and address some of these questions, several years ago now, my lab performed a genome-wide screen for novel regulators of myofibroblasts. And we actually did this in a fibroblast where we transfected over, I'd say, 1,800 different cDNAs for different human and rodent genes. And then, oh my goodness, and then we screened for whether or not they turned on the myofibroblast program using a transcriptional surrogate shown here. And this transcriptional surrogate is really just a simple luciferase reporter that was driven by the alpha smooth muscle actin promoter. And so down here is is a graph of from one of the plates from this high throughput screen that we performed, where you're looking at luciferase activity as a function of well number, and of course in every different well was a different cDNA. And one of our most dramatic hits or significant hits was a cDNA for a gene called muscleblind-like-1 or MBNL1. And so some of you may have heard of MBNL1 before because it's highly related to myotonic dystrophy. And in fact, when it's dysfunctional, it causes severe muscular and neuromuscular defects. But at the time when we identified this gene, we had never heard it being associated with wound healing or the heart or the fibrotic response. So this was a really intriguing hit. So what does muscleblind do? Well, it's actually an RNA binding protein, and what its function is to really mature the transcriptome. So it can alter its target transcripts polyadenylation. It can control the splicing of its targets. It can control its stability of its targets, or it can control where the transcript is localized within the cell to either promote or detract from translation. So it's a pretty powerful molecule that can have a major effect really on the transcriptional identity of a cell. And so one of the things we wanted to do first was just to say, okay, maybe this is like a phenomenologic gene that we pulled up in a screen. Does it have any physiologic relevance to heart disease or the fibrotic response? And so in collaboration with April Stenfiano-Turo, we got cardiac fibroblasts from healthy and failing human hearts. And then we just looked using RT-PCR to see whether there was a change in gene expression of muscleblind in those two cardiac fibroblast fractions. And here's the results from that experiment. What you can see is that there was a significant upregulation of muscleblind activity in fibroblasts from failing human hearts that when you compared them to healthy. And so right away, we were pretty intrigued. This might be a bona fide hit. Weren't really satisfied with our understanding of its relevance just from that experiment alone. So we undertook a lineage tracing approach to really try to identify if muscleblind activity changes in some of these important fibroblast populations. And so in this particular experiment, what we did was we took a lineage reporter mouse for myofibroblast, and that lineage reporter is driven by a periostin Cre driver. And so in this particular Cre, a tamoxifen-inducible Cre cassette is knocked into the periostin locus, and then we cross that mouse line with one that has a fluorescent reporter on it. And so basically, the offspring from these mice will label any cell that has become an activated myofibroblast with a permanent green fluorescent protein. So that allows us to track these cells over time in the heart. And so in this particular experiment, what we did was we took those myofibroblast lineage reporter mice, we subjected them to myocardial infarction, and at several time points following myocardial infarction, we looked at the fibroblast population for muscleblind expression or muscleblind activity. And what we did was to even further refine the myofibroblast or the fibroblast population by taking out the entire cardiac fibroblast population and further segregating it into those who are inactive or quiescent versus those that were labeled green and our lineage trace for being a myofibroblast. And so the results of that experiment are shown for you here. And what you're looking at is muscleblind expression as a function of time following myocardial infarction. And what you'll notice is that in our periostin lineage trace cells, we saw significant upregulation of muscleblind expression at both days 4 and 14 following myocardial infarction. And we were really excited to see that this actually tracked with the expression of canonical myofibroblast markers like smooth muscle alpha actin shown here, as well as collagen 1A1. And so now we were feeling pretty good that this is a relevant target from our screen. So we went on to model this change in muscleblind expression with a transgenic mouse. And we wanted to ask the question, well, is muscleblind expression alone sufficient to make a myofibroblast and sufficient to create fibrosis in an otherwise healthy heart? And so to create that model, we developed a conditional muscleblind transgene. And so this is a Cre-inducible transgene. And then we crossed it with a quiescent cardiac fibroblast specific Cre that's driven by knocking a Cre cassette into the TCF21 locus. And if you'll recall, TCF21 labels the resident cardiac fibroblast. And so we created these mice, we let them develop normally, so the fibroblast population should have developed, and then we induced the expression of the transgene with tamoxifen once these mice were fully developed. So 2 weeks after tamoxifen induction, we excised the cardiac fibroblast population and just used RNA-seq analysis to say, okay, what do these cells look like transcriptionally? And the results of that RNA-seq analysis is shown here. And what we found was that the fibroblast from the transgenic mouse was completely enriched for, again, canonical myofibroblast gene expression. And those genes include an upregulation of ACTA2, lysyl oxidase, which is a cross-linking enzyme. Several of the collagens were upregulated. And of course, we saw an upregulation of periostin and fibronectin, which is a classic hallmark, again, of the myofibroblast population. So we also wanted to do a functional assay to confirm that these cells are actually functionally transitioning from a quiescent cell to an activated state. And the way we do this assay is we take cardiac fibroblasts out of these mouse hearts, we put them in two-dimensional culture, and then we treat them with a pro-fibrotic agonist like TGF-beta or angiotensin 2, and then we score for the number of cells that become alpha smooth muscle actin positive. And so just to show you what a normal fibroblast response looks like in this assay, you can see that here's a control population or a non-transgenic population of cardiac fibroblasts, and there's very little alpha smooth muscle actin stained cells. And in this particular assay, alpha smooth muscle actin is shown for you in red. But if you compare that to the TGF beta treated population, you can see it's littered with red stained cells. And in fact, with TGF beta, you get about a 60 to 85% conversion of the cardiac population, the cardiac fibroblast population with that treatment. Now, if we compare it to our muscle-blind transgenic cardiac fibroblasts, you can see that even in the absence of a profibrotic agonist, these cells were already primed or converting to that myofibroblast phenotype or to having alpha smooth muscle actin positivity. And then when we treated those cells with TGF beta, we really didn't get much of an additive response. And so that suggests that just by having the transgene alone, these cells are already pushed all the way over to that differentiated state. And those data are quantified for you here. So I think the big question was, well, did we make fibrosis in these animals, right? We turned on this gene program. Did anything change at the organ level? And I can tell you that at the 2-week time point, when we made most of these measurements of cell phenotype, we didn't see any fibrosis. But if we let these mice age a couple more months, we saw an accumulation of interstitial fibrosis, which is again quantified for you here. So this suggests that indeed muscle-blind is sufficient for not only transforming the fibroblast into an activated cell type, but for creating fibrosis in an otherwise healthy heart. Oh my goodness. Okay, so in this experiment, we really wanted to challenge these mice with the expectation that if we gave them an injury like myocardial infarction, they should have a robust and wicked fibrotic response, that they really should do poorly. And so what was really surprising when we did this was that they didn't do much worse, at least in terms of the fibrotic response, than their non-transgenic litter mates. So in this particular experiment, we subjected both the non-transgenics and the muscle-blind transgenic mice to myocardial infarction. 2 weeks later, we measured their fibrotic scarring, and this is the outcome from that measurement. You can see there's a little bit of a difference in fibrosis, but it really wasn't, I mean, significant or overwhelming. Definitely not what we expected when we did this experiment. We did look a little bit more in depth at the maturity of the collagen, and we can do this using simple imaging methods where we measure the birefringence of collagen using polarized light microscopy. And we did see that the scar was way more mature, but we definitely weren't getting more of it. And so that was just a surprising finding to begin with. So one hypothesis we had regarding why we wouldn't have seen more scar is that when we turn this transgene on, we turn it on before the injury. And there's a really critical function that happens following myocardial infarction in a fibroblast, which is before they differentiate, they wildly proliferate. And so you get a lot of expansion of the fibroblast population before they turn into these scar-forming cells. And so one of our hypotheses was, well, maybe this transgene actually impacted the first wave of proliferation. And so the actual cell number was reduced, even though they're more active. So we went into these hearts and we measured cardiac fibroblast proliferation by treating these mice with EDU. And EDU is a chemical that incorporates into proliferating cells. And then you can use histology to screen for those cells. And so what we found was that when we measured proliferation, and that data is shown for you here, we saw a significant decrease in the number of PDGF receptor alpha positive fibroblasts that were also EDU positive, telling us that the population was indeed impaired for proliferation. Okay, so as cardiologists, I hope you're wondering, well, how did the heart function in these animals? And so this is some really, I think, from all the echocardiography we did on these mice, the main finding was they do really poorly from a standpoint of systolic function. So if you look at fractional shortening here, you can see that their systolic function is decreased. And this again was a little bit surprising because we really didn't see a greater amount of fibrosis, only an increase in maturity. So we were surprised that it had such a profound impact on the shortening of the hearts. We also saw that these hearts were dilating more, and we saw this at a morphological level in the myocytes. So we actually digested these hearts, measured the length and width ratios of isolated myocytes. We found that these myocytes were more narrow as well as longer, and that's a classic morphological feature of dilation. So the hearts don't do very well. Okay, and in this experiment, we really wanted to then address how our transgene was impacting the heterogeneity of the fibroblast population. And so the way we did this experiment was to use single-cell RNA sequencing. We did single-cell RNA sequencing of just the total cardiac fibroblast population that was flow sorted out of non-transgenic animals and muscle-blind transgenic animals in a sham and infarcted condition. And so just to orient you to this data, I want to first focus on this hierarchical clustering, a UMAP plot shown here on the left. So this is the hierarchical clustering of every single cell we sequenced, independent of their genotype or the experimental condition. And each color represents a different fibroblast cluster that's based off of gene expression, differential gene expression. And so first, just to look at the whole plot in general, we found 13 different states or different types of fibroblasts in the total cardiac fibroblast population. And they seem to segregate into three sort of larger what I would call differentiated states. So we found that there was a section of highly epicardial like cells that were shown for you here on this plot. We found our quiescent cells, or quiescent fibroblasts that were really defined by a high expression of PDGF receptor alpha and TCF21. And then we had this group or cluster of cells that we really call activated myofibroblast states that were defined by an upregulation of periostin. And within those myofibroblast states, we really saw three prominent substates, and using pseudotime analysis, we found that these states shown here in green and blue are sort of intermediate states. They're not fully matured yet, but still are expressing periostin. But once a cell hit this pink state, or our Myo1 state, they were in their most mature phase, where they're expressing alpha smooth muscle actin. And so this was a really nice way for us to see kind of how these cells, or the trajectory of these cells during the differentiation process. And so now I want to focus on the data over here just to show you how the different genotypes segregated under the different experimental conditions. And if we just look at our sham condition and compare the non-transgenic to the muscle-blind transgenic, what you can see is that the muscle-blind transgenic without any injury was getting, was starting to have cells that live in this state space of a myofibroblast. And you can see that in a non-transgenic, that was virtually absent. And then when we give an MI, you can see that the non-transgenic mice really have a full diversity, or they have an increased diversity, where they have fibroblasts sitting in both quiescence and all of the activated myofibroblast states, whereas the muscle-blind transgenics were really pushed towards that most activated and mature state. And so I think this is a really nice demonstration from a transcriptional standpoint of how this transgene really impacts the program differentiation of the cell type. It's not that we created a new cell type, we just kind of pushed it along its normal differentiation trajectory. Okay, so one of the next questions we asked was, well, what muscle-blind regulated transcripts does muscle-blind control in order to create this myofibroblast transition? And so again, we modeled the gain in muscle-blind function by using adenoviral overexpression of muscle-blind and cardiac fibroblasts. And then we used RNA immunoprecipitation to segregate all of the RNAs that are bound to muscle-blind in a cardiac fibroblast. And then we identified those that subset of transcripts by RNA-seq analysis. And so what we found from this assay was about 2,500 transcripts are actually bound by this protein. and that most of those transcripts are enriched for the cell biological processes of differentiation, and more importantly, mesenchymal cell differentiation. So this is really exciting because it fits kind of all of our stepwise data that we've seen that I've shown you in previous slides. This is just a validation of that RNA precipitation analysis. And really what we're showing you here is just RT-PCR validation. And if you look at the immunoprecipitation lanes from our control group versus the ones that are overexpressing muscleblind, you can see that muscleblind's pulling down transcripts for genes like TGF-beta receptor 2, calcineurin, serum response factor, and ROCK2. And when we did further analysis of this, we found that muscleblind was stabilizing TGF-beta receptor 2 and SRF, and then alternatively splicing calcineurin and ROCK2 to create a constitutively active variant. And so basically what was happening is we were getting a gain in function of all of these different pathways. What I haven't told you is that in previous work, we had identified that all 4 of these factors are really nodal regulators of myofibroblast differentiation. And so from this data, what we think is happening is muscleblind is sort of promoting an entire cell differentiation program. Okay, so this is an experiment we performed really to ask the question, well, is muscleblind essential? So we show that if you overexpress it, it's sufficient, but do you really need it to create a myofibroblast and get fibrosis? And so the way we addressed this question was to create a conditional muscleblind knockout mouse. And we crossed it again with our cardiac fibroblast specific CRE that's driven by the TCF21 locus. And then we took these mice and gave them tamoxifen following development and subjected them to a myocardial infarction. And again, we looked at these mice about 14 days later. So here's the results of that experiment. And what you'll notice is we had a significant decrease in fibrotic scarring in these mice. And when we looked at the maturity of the scar, again, using collagen birefringence imaging that's shown for you here, we saw that the fibers were highly immature. So this was a really weak scar that formed. And luckily, we didn't have any cardiac rupture in these mice. We also scored for the number of myofibroblasts that we saw in the tissue by counting the number of alpha smooth muscle actin positive fibroblasts. And you can see here we saw a significant decrease in the number of myofibroblasts that had formed, at least histologically, in these hearts. Now, given that we saw a profound difference in proliferation when we overexpressed the transgene, we also looked at proliferation in our knockout mice using the same approach where we gave EDU to these mice and then looked at the peak of proliferation following myocardial infarction. And so the data from this experiment is shown for you here. And what you can see is we saw a significant increase in the number of fibroblasts that were positive for a cell cycle marker. And so this suggests that indeed these fibroblasts are more proliferative. We drilled down a little deeper and we pulled the entire cardiac fibroblast population out of the hearts and subjected them to RNA-seq analysis. We found supporting evidence for the proliferative phenotypes in which we saw an increase in proliferation genes like CDK6, cyclin D1, and then probably more importantly, we saw a significant downregulation in a really potent cell cycle inhibitor, which is CDKN1A, which is also P21. And in further analysis, we found that muscleblind actually binds and regulates CDKN1A. So we think this is a direct conduit to why when you have gain or loss of muscleblind function that you have this change in cell proliferation. Okay, so how do these mice fare following myocardial infarction? Well, the muscleblind knockout mice do quite a bit better. You can see they have improved global lateral strain shown here. And when we measure dilation using echocardiography, they're less dilated. So they definitely do better when you prevent this myofibroblast transition. So this is a follow-up experiment that we wanted to do basically to ask the question whether or not you could reverse the myofibroblast phenotype, or you could let these mice have a myocardial infarction and then change their phenotype following, because of course we won't be able to predict when someone has a heart attack. So the way we approached this question was, again, to use our conditional muscle-blind knockout, only we crossed these mice with our periostin-cree. So what that allows to happen is that these cells will begin the differentiation program, and then it's about 3 or 4, I should say about 4 or 5 days following myocardial infarction that the muscle-blind gene will actually get excised from these cells. So it really allows them to kind of feel the injury first before we change the program. And so the results from this experiment are shown for you here. And what you can see is that we still can impact the fibrotic response. It's significantly decreased. Even if you take muscle-blind out following myocardial infarction, we actually get a better improvement in fractional shortening, as well as a better rescue of dilated remodeling. So it seems to me from this study that there's a little bit of positive effect of a myofibroblast at first. You just don't want to keep them around too long. And since, just in the interest of time, because I want to save time for questions, I'm going to skip these next 2 experiments for you here. Okay, so again, we looked at the heterogeneity of the population, only this time following muscle-blind knockout. We set this experiment up in the exact same way as the single-cell experiment I showed you in previous slides. And I think this is really the part of the figure that you want to pay attention to, in which you can see that when we take muscle-blind out of the system, we completely lose every single activated state, and that these cells are just really sitting in total quiescence. And so that kind of fits with all of the data I've shown you, at least from a functional level. So with that, I'll take any questions.
Thank you.
Yeah.
For that really great talk. Lots of questions here. We'll start with
Thanks, Dan. That was great. So 2 questions. First one is about the mechanism of action. And you seem to allude to the fact that muscle-blind is binding and stabilizing the RNA of various transcripts.
Yep.
Do you know much about that binding? Is it all in the same place? Is that a drug target? Because it seems like inhibiting, you wouldn't have to knock down muscle-blind. You would just simply need to block that stabilization.
Yeah, so the stabilizing function we think mostly comes from target sites in the 3' UTR, and there's no putative sequences that it binds to. So you could actually, if you want to think about a therapeutic, you could probably create a little oligo sponge that would outcompete the natural binding sites if you wanted to knock it down. Yeah.
And you didn't touch on it, but I would just be interested in your thoughts. You started from a quiescent fibroblast, but you didn't touch on where those quiescent fibroblasts came from. So I would appreciate your take on the data.
So do you want to know developmentally where those come from? Yeah.
Or if the population that is muscle blind is unique.
Yeah, that's a good question. So the fibroblasts we were studying are epicardial derived fibroblasts, although I do believe endocardial derived fibroblasts can express TCF21. However, we probably do a better job of getting all of the developmental lineages when we target muscle blind using the periostin-cree, because all of those lineages will ultimately express periostin. And then the TCF21 lineage that's epicardial derived misses pretty important areas of the heart. So like you don't get fibroblasts in the septum, you don't get some of the fibroblasts in the right side of the heart. So we have patches that we're not targeting. Does that answer your question? Okay. Yeah.
Jen, that was just a fantastic talk. I have 2 questions. You presented a majority of your data are RNA sequencing.
Yeah.
But the exciting and unique part was at the end. I'm assuming, are you guys doing spatial transcriptomics to try to merge those 2 data sets? And is that amenable to the type of staining that you're doing? That's question number 1.
Okay, that's a good question. I feel like reviewers, when we submit this paper, are going to ask us to do spatial transcriptomics. The problem is the resolution of spatial transcriptomics at this point for in vivo imaging is not at cellular resolution. It's too big. So the other problem is that most of the tissue preparations you have to use for spatial, it quenches our lineage reporters. So we could do it, but it just, I don't think would be as good. So we've talked to Jay Shanduri about how to use some of his methods. They've never tried their methods in vivo, so that would be a huge technical challenge, but I feel like we're going to have to do that.
Yeah, I assumed that was the case, but I wasn't sure. Second question is, I was really impressed by the end of the talk. Can you comment about, do you see any clinical utility to that kind of assay? I mean, obviously, I know the data and I know it is very recent.
Yeah.
Within, so you haven't had probably a lot of time to think about it. Can you comment about, is there any clinical utility in that kind of scoring system, or have you had any thoughts on that?
Oh, you and I have talked about this already. Yeah, I mean, yes, I mean, you could definitely see places where you could use imaging for clinical diagnosis or to develop prediction pipelines. Maybe Pat can help us with that in the back there. So yeah, I mean, we're so new. It's such new data that it's hard to know what to do with it.
Yeah, thanks for a very interesting talk. We've got a couple of questions. I thought I read once that TCF21 is expressed by pericytes, and whether that might interfere with the instrumentation center of the data, particularly when you start to talk about interaction with vessels.
Yeah. So TCF21 is expressed during the lineage program, but once a pericyte develops, it does not express TCF21 anymore. I was afraid that we were imaging pericytes, and so we actually went back and did pericyte imaging, and there's no overlap between the TCF21 and the pericyte.
The next question is, with the cluster, the single cell analysis and clustering, ideally one would get the sort of discrimination that you get in construction. A house is being built, but then you identify the plumbers, the electricians, people pouring the foundation. How far are you away from assigning roles to these various populations?
I think we're really far, to be honest, because to really assign those roles, we're going to need targeted genetic approaches. And when you look at the clustering, they all have overlapping genes. So you just see sort of, it's the amount of the gene expression that defines their clustering, but it doesn't mean the gene is absent in those different clusters. So finding sort of a unique target for each of the clusters, I think is going to be extremely challenging. But we'd love, that's like where we're headed. Might need more deep sequencing, right? Like really deep.
Hey, Jen, great talk on a great arc of work. Two related questions. Have you imaged the role of myofibroblasts or MBNL1 in and around sinus node, AV node, and His-Purkinje tissue? Then the related question is, you mentioned MBNL1's role in myotonic dystrophy. MD1 at least has cardiac manifestations, which seem to be accelerated in the conduction system.
Mm-hmm.
So is there a MD1 biobank that you could utilize to make such a connection?
Oh, that's a good question. For the last part of your question, I don't know if there's a biobank on that. We have not yet looked sort of at electrical, that side of things, but the data is there, so we could definitely do that.
So I have a follow-up electrophysiology question.
Oh no.
So have you looked at, or is there an implication for better understanding atriopathy?
So I'll tell you, we have not fully investigated the atrial phenotype, but there's a huge atrial phenotype in these mice, and it's actually worse if you specifically knock muscleblind in and out of the myocyte. So we have some really nice models. We just don't know enough about the atria. So if any of you guys who know more about the atria want to join us, we could use your expertise, because they have a huge phenotype, but we don't know we usually throw atria away. So we could use your help. Yeah. Did I pop those up? Okay, they can do it. Okay, great.
You can take it if you see.
Interesting.
I have a question from April Stampion. Hearts after myocardial infarction have essentially 3 different areas: scar, border zone, and uninjured. Were you able to find differences in ssRNA or vessel associations in the different areas?
The morphology is definitely different based on their anatomical location relative to whether they're in the scar, on the border zone, or in the remote zone. So we found that. With respect to vessel interactions, we did not see, well, let me back up. There, of course, is a difference in the middle of the scar versus the border zone, because the middle of the scar has very little vasculature. So there's very little chance for fibroblasts that have migrated in there to interact with the scar. But on the border zone, we have robust interactions. If that answered your question, April. Anything else? Okay.
Okay.
Thanks,
so much for the
everybody.
great talk. Thanks everyone for the great questions. Have a good day.
Yeah.